Today's lecture, we're going to talk about the detection of blood at a crime scene, and we are going to finish up with the other bodily fluids that can be used for identification. So the first thing that they have to figure out is if it really is blood at the crime scene. There's a couple prelim color tests they can go through. Benzidine is actually a carcinogen, which means it causes cancer, so it is not used as often. But if you put that together with the potential blood stain and hydrogen peroxide, it will turn a pink color. The one that you watched a video on was the Kastle-Meyer test, and that includes phenolphthalein, which also turns a pink color when mixed with hydrogen peroxide. The luminol test reacts with blood to basically glow or produce a luminescence. And this one's really good because it is sensitive to blood that may have been diluted, aka cleaned up. And it is bad or potentially bad because it can destroy many of the important blood factors. The last one, called a microcrystalline test, the hemoglobin in the blood will form a characteristic crystal, but it's a lot less sensitive and it's a lot more susceptible to interference from other contaminants from the crime scene. So here you see the luminol test being happened with the blood glowing. Now you figured out it is blood, so is it human blood or animal blood? And what happens is they draw blood from a human vein, inject it into a rabbit, and then what the rabbit does is it sensitizes to the blood, and it can kind of not react with the blood, but it ends up forming a precipitin that says, hey, this is not rabbit blood, it's not human blood, or excuse me, it's not animal blood, it is human blood. All right, so requires only a small amount of blood, and it can work on dried blood stains as old as 10 to 15 years, which is awesome. So basically what we're doing is we're creating a precipitin that says, hey, this is not animal blood, it is human blood. Another way to do that is something called gel diffusion. This is very, very similar to gel electrophoresis, which will be discussed on the next slide. Agar is basically a jelly-like substance, and we're going to put the blood stain and human antibodies in the jelly, and if they move towards each other and form a line of precipitation, which is right there, it is human blood. Same thing, what would happen here is the blood and the antibodies would go in these wells right up here. Electricity would then be run through this gel, and if the antibodies and antigens move towards each other, it would be human. The last concept we're going to cover is something called serology, and that's the study of serum. So there's serum and blood, as well as other bodily fluids, and these are used for identification of different individuals. So saliva is a mixture of water, mucin, which aids in swallowing, amylase, which is used for digestion, and buccal cells, which are cheek cells, and those are a really, really good source of DNA. And these can be associated with a sexual assault or bite marks. Semen is composed of water, sperm, enzymes, and salts. And this can be tested for in a couple different ways. All of the presumptive tests include being able to fluoresce or again glow under UV light, or it will also turn purple in the presence of acid phosphate. As far as confirmatory tests, there would be microscopic examination of the sperm, or it could be used for DNA typing. Urine can be used for detecting drugs using the emit technique, and there's antibodies that will bind to specific drugs added to a urine sample.